Hello everyone, I'm TURKIYA and today I would like to talk about this article. Phosphoproteomic analysis of induced resistance reveals activation of signal transduction processes by beneficial and pathogenic interaction of gray vein. This article is written by Michel Brazzoli and his colleague from Department of Sustainable Agro, Ecosystem and Bioresources in Italy. The aim of this study is to identify mitogen-activated protein kinase that are phosphorylated and dephosphorylated during grape vein response to the pathogen Plasmopara viticola and to the beneficial microorganism Trichoderma herzinum T39 in order to get an overview of proteins and mechanism implicated in T39-induced resistance. Grape vein is a major fruit crop worldwide, and it can be attacked by a large number of pests and pathogens. Biotrophic omicide Plasmopara viticola causes grape vein downy mildew, and this requires frequent application of chemical fungicides to avoid yields and quality loss. Beneficial microorganisms have been shown to induce grape vein resistance to downy mildew, and they represent promising alternatives to chemical fungicides. The plant immune system recognizes exogenous microorganisms and responds by activating defense mechanisms through phosphorylation and dephosphorylation events within the proteome. Mitogen-activated protein kinase and phosphatase cascades are important for transducing signals generated by receptors into cellular responses through the downstream phosphorylation changes of target protein. These processes are essential to establishment of pathogen resistance. In the next slide, I will present the material and methods of this study. First step was resistance induction and pathogen inoculation. 2-year-old plants were grown for 2 months under greenhouse condition at 25 degrees. All leaves of grape vein plants were treated with water or T39. A fresh suspension of P. viticola was sprayed onto apical leaf surface of grape vein leaves and incubated overnight. 10 days after inoculation, disease severity was assessed visually. The second step was sample collection and protein extraction. Leaf samples were collected immediately before inoculation at 0 hour, at 6 hours, and at 24-hour post-inoculation in triplicate for each treatment. For protein extraction, ground leaf samples were suspended in 10 volumes of trichloroacetic acid in acetone solution for overnight precipitation. Protein concentration was determined with a biorad protein assay reagent. Immunoplot analysis step. Protein samples were subjected to SDS page. Total and phosphorylated mitogen-activated protein kinases were detected by overnight incubation at 4 degrees centigrade with antibody raised against the extracellular signal-regulated kinase 1 and 2, or with the antibody raised against phosphorylated extracellular signals-regulated kinase 1 and 2. Signals intensity of 45 and 49 kilodalton bands corresponding to grape vein mitogen-activated protein kinases was quantified by the quantity 1 4.6.1 software biorad. Immunohistochemical localization analysis. The third and fourth leaves from the top of each plant were collected and treated with fixative solution of 5% glutaldehyde in phosphate buffer solution. Histological observation were carried out with LMD 7000 microscope. Protein digestion and labeling for iTrack step. Protein were digested twice with trypsin. The digested peptides were labeled with 2 8-plex iTRAC reagents. One contained protein extracted from water-treated plants at 0 hour and at 24-hour post-inoculation, and T39 treated plants at 24-hour post-inoculation. The second one contained protein extract from water-treated plants at 0 hour and T39 treated plants at 0 hour and 24-hour post-inoculation. Peptide fraction and phosphopeptides enrichment. The iTRAC labeled peptide mixture was fractionated by strong cation exchange chromatography. Enrichment phosphorylated peptides was obtained by sequential elution protocol. 4 elution from immobilized metal ion affinity chromatography, and 4 elution from titanium dioxide. The 8 elution were acidified with TFA, clarified with C18 starch tips, and subjected to liquid chromatography, linear trap, quadruple, orbitrap mass spectrometer analysis. Liquid chromatography tandem mass spectrometry analysis and identification of phosphopeptide. Each sample was analyzed by an easy LC capillary chromatographic system connected to linear trap, quadruple, orbitrap mass spectrometer equipped with a nano electrospray ion source. The mass spectrometry proteome data have been deposited to the Peptide Atlas database. Raw data obtained from the liquid chromatography mass spectroscopy runs were processed with the Proteome Discover software, which uses data from the mascot search and performs relative quantification of the 8 receptor ions derived from the iTRAC reagents. Integrated approach for identification of quantified protein. Protein identification was carried out by mascot search against 3 protein databases: the Pinot-Noir grave vein proteome, the NCPI grave vein proteome, and the non-redundant Viridiplantay UniProt protein database. Identification of peptides with significant changes in phosphorylation. The iTRAC ratio of each identified peptide were logged to transform to generate a normally distributed set of data and processed using the multi-experiment viewer software. Volcano plot visualization of MEV software were applied to identify peptides with significant change in phosphorylation. The following slides show the results obtained from this study. The experiment was repeated 3 times with similar results. Downy mildew severity was significantly reduced by application of 339 compared to water. In water-treated plants, the abundance of total 45 and 49 kilodalton mitogen-activated protein kinases significantly increased with P viticola at 6-hour post-inoculation, figure A, and at 24-hour post-inoculation, figure B. The abundance of the total 45 and 49 kilodalton mitogen-activated protein kinase was increased by T39 before P viticola inoculation at 0 hour, and it was not further increased after P viticola inoculation. T39 treatment increased the phosphorylation level of 45 and 49 kilodalton mitogen-activated protein kinases before P viticola inoculation in figure A and B, and the high phosphorylation levels was maintained at 6 and 24-hour post-inoculation. Moreover, the level of the phosphorylated 49 kilodalton mitogen-activated protein kinase was significantly greater in T39 than in water-treated plants at 24 post-inoculation. This figure shows the results from immunohistochemical analysis. The phosphorylated mitogen-activated protein kinases were not detected in water-treated plants at 0 hour as shown in figure B. While in T39 treated cleaves, strong fluorescence of phosphorylated extracellular signals regulated kinase 1 and 2 hybridization was detected in T39 treated plants at 0 hour. That reflects a direct pitogen-activated protein kinase activation before pathogen infection, as shown in figure F. The phosphorylated mitogen-activated protein kinase were activated at 24-hour post-inoculation in water-treated plant. Fluorescence intensity of total and phosphorylated mitogen-activated protein kinases was greater in T39 than in water-treated plants at 24-hour post-inoculation. The integrated approach of mascot search against 3 different databases was developed to improve protein identification and annotation, and it allowed the identification of 109 grave vein phosphopeptides in ITRAC-1 and 56 grave vein phosphates in ITRAC-2 experiment. 56 phosphopeptides were assigned to grave vein protein consequence by searching against the Pinot-Noir grave vein proteome. 54 phosphopeptides were assigned to protein sequence of the NCPI grave vein proteome. 55 phosphopeptides were assigned to protein sequence of the Viridiplantae UniProt database. Among the protein identified against the NCPI grave vein proteome, 39 matches with protein of the Pinot-Noir proteome and 15 with no homology. Among protein identified using Viridiplantae UniProt database, 9 homologous with protein of Pinot-Noir and 19 homologous with protein of the NCPI grave vein proteome. The 165 phosphopeptides corresponds to 137 protein of the grave vein genome database and 28 novel and predicted grave vein protein. This figure demonstrates the distribution of amino acid phosphorylation in phosphopeptides. The distribution of phosphorylation sites identified 165 phosphopeptides was 61% on serine, 30% on threonine, and 9% on tyrosine. And this figure shows Venn diagram of peptides with significant changes in phosphorylation. Peptides with significant changes in phosphorylation were identified by 4 pairwise comparisons between grave vein treatments. 44 phosphopeptides were affected by P. viticola inoculation in water-treated plants. 21 phosphopeptides were affected by T39 treatment. 51 phosphopeptides were affected by P. viticola inoculation of T39 treated plants in comparison to water-treated plants at the 0 hour. 31 phosphopeptides were affected by P. viticola inoculation of T39 treated plants at 0 hour. This figure shows an overview of grave vein proteins with significant changes in phosphorylation in response to T39 and P. viticola. The overview of cellular process highlighted signals transaction pathway and defense response affected by phosphorylation changes during interaction with T39 and P. viticola. Different recognition pathways were activated by P. viticola in water and T39 treated plants. Results for specific phosphorylation changes of T39 treated plants in response to P. viticola are 9 peptides show opposite phosphorylation profiles after P. viticola inoculation in water, and T39 treated plants such as a mediator RNA polymerase and disease resistance protein. They may contribute to the modulation of infection and resistance related process. However, some proteins implicated in microbe recognition and phosphosynthetic process showed similar phosphorylation profiles after P. viticola inoculation of water and T39 treated plants. In summary, activated protein kinases was increased by T39 treatment before pathogen infection, and it was maintained at high level after P. petechiae inoculation. Mitogen activated protein kinases were activated by T39 in absence of the pathogen, and they could be involved in the signal transduction cascade to pre-activate grape resistance mechanism. The phosphoproteomic analysis identified 103 phosphopeptides with significant changes in phosphorylation in response to beneficial and pathogenic interaction. Should this paper have been published? Yes, it's good to publish this paper as it investigates an interesting and very specific topic. And it provides helpful information and good recommendation for future research. It presents good data analysis, and there is no ethical issue as the experiment was performed on plan. Thank you.